University of Science and Technology. It's my great pleasure to invite today's speaker, Dr. Brodie. Seth Brodie obtained his PhD in biochemistry from the State University of New York at Buffalo in 2005. After this, he just got a couple postdoctoral programs with the first in Emory University. And then he moved to NIH when he focused on the functional population genomics of rare cancer, predisposition syndrome. And Dr. Brodie was one of the first people to work with the better version of the Levy cell system. Now he's working in the Levitas Bio as an associate director of research and development, where he's focused on application development surrounding single cell genomics. Saf today is going to talk to us about using magnetic levitation as a label-free sample preparation method for functional and single cell genomic applications. Dr. Brodie, the floor is yours. I would like to ask all of the audience to turn off their mics and videos for better streaming. And if you guys have any questions, please write it down in the chat. I will ask Dr. Brodie to stop the talk and answer the question when it's needed. So all yours, Saf. Thank you.
Thank you very much, Mohammed. You're really a nice guy to invite me to give a talk to you guys today.
You're most welcome.
I'm very excited to talk about my work that's been going on at Levitas for the past 6 months. Since I moved to Levitas in April, we've been developing new applications for our system, which uses magnetic levitation for sample preparation. So I'll go ahead and get started. Thank you very much for your attention. Okay, so today I will be talking to you, give you a bit introduction to the system itself, which is completely label-free, so you don't have to perturb the cells at all to separate them. Then I'll talk a little bit about single-cell RNA sequencing using the 10x Chromium system in line with our Levicell system. And then I'll talk about a project that is still underway with collaborators at University of California in San Francisco that is using CRISPR and Levicell using CRISPR. And then we'll conclude and I'll answer some questions if you have them. So whenever you're doing any sort of functional work, genomics work, the most important thing is the sample you're working with. So if you have, if you're working with samples from patients, blood or tissues or animals, or plant materials or cells that you've somehow engineered either with CRISPR or with other protein engineering methods, the most important thing is the quality of your sample. And very often what happens is you have this rare sample and the quality is so low that you can't really use it. And so there's high numbers of samples that are just stuck in freezers at universities and institutes all across the world that can't be used because their sample quality is so low. And so what we're able to do is to break that bottleneck and take these failed and precious samples that would normally not be able to be used for sequencing or for any sort of functional analysis, and we can harness those samples. And so, you know, if you have a poor sample and you lost the time, you lost the money, not able to use these samples. And so now we can use these samples. And so that's the key to our the Levicel. So one other aspect of ways people are currently getting usable samples from these poor starting points is doing all sorts of different methods. They either use a FACS sorter, or they use magnetic beads, or they use harsh centrifugation. All of these have pitfalls. They change the cells themselves. They're harsh. You wind up label-free and enriches the sample, and at the end you have a pristine sample. So our system, the Levicel, works on the process of magnetic levitation. Now this sounds like magic almost, right? But it was a technology that was invented by George Whiteside's lab at Harvard, and then further developed by a group at Stanford University that then spun off into our company. So our company's been around for about 3 years now, and we have this commercial product called the Levicel. And so the Levicel uses a magnetic field to create a virtual density gradient that allows cells to be separated without altering them at all. So you can use this for drug analysis, to look at multicellular organisms like Plantera or C. elegans. You can use it for cell suspensions. You can use it for plant materials. You can use it for yeasts. Really, as long as it's below the upper size limit of 300 microns, you can put it in the system. So how does it actually work? Well, there are two very strong magnets that are separated by a small channel, and the cell suspension or whatever your material is resuspended in a paramagnetic medium, which is similar to the agent that's used for MRI, for magnetic resonance imaging. So your sample is resuspended in this buffer, and it allows the cells to float in just based on the inherent buoyancy of the material. So you can think of it sort of as like you have cells that are similar to like a beach ball in the ocean. And so the ball excludes the ocean water, and it prevents and it floats in this neutral buoyancy manner. And so as the cells are levitating, they levitate to a place of neutral buoyancy, whereas particles that either absorb the buffer around it, or debris, or dead cells, they sink to the bottom of the channel. Here's another graphic to illustrate this. So you have these two magnets that are facing opposite, similar poles towards each other, and it creates a magnetic field that allows for density gradient across this magnetic field. And an additional aspect of our system is that it has real-time imaging capability with both red and green fluorescent channels. So although it's not critically necessary for cell separation, you can fluorescently label your materials, and then you can image them in two different channels plus bright field. And so you can have any sort of starting material from any source using any sort of buffer. All you need to do is spike in the levitation agent, and it allows for the levitation to occur. It's right now, most of our applications are surrounding viable cell enrichment. So separating pristine live cells from dead cells, dying cells, and debris. So in this graphic, the green and yellow balls are live cells. The red are dead and dying cells. The gray is debris, and blue is molecular background, so free RNA, free DNA. And what happens as the material sits in the magnetic field, it separates just due to the inherent buoyancy of the particles themselves. And so the dead cells tend to sink like anchors. The live cells tend to float like the beach balls, and the materials that are separated in this way. And so in development are further applications that allow for depletion of contaminating particles. So we can get rid of certain cell types that we might not be interested in just by anchoring them down to the bottom of the channel. And also in development are targeting assays where we can pull our favorite cells up to the top of the channel. And this way we can separate cells exactly how we want them. Also, what we're able to do, and that's currently still in development, is actually onboard characterize the cell populations. So we can do some cell counting assays or viability counting. We'll be able to look at different cellular states, looking at sample purity and quality, or any other sort of aspects of the cell. And so our system, what it does is it's a single, it uses a consumable. So you don't have to wash the system like in a fax sorter. It's very easy to use. It's gentle to the cells. It's flexible. You can use any type of particle, and it's very, very efficient. So how does it actually work? You take, let's say you're using cells from a cell culture, and you just take your cells, you count them, and you resuspend them in our buffer, in any buffer that contains our levitation agent, and then you load it into the cartridge and you put it in the system, and about 15 to 20 minutes later, your sample is ready for collection. And then you can do a host of things downstream. Single cell sequencing is one application that we do quite often, but there are many functional assays that you can do, or you can continue to culture the cell. So looking at multiple passages and maybe studying how cells change over passage time. All of these things are possible with the system. And so, as I mentioned before, you have this single-use cartridge. So you load your sample in the cartridge, and it's you can get a sterile cartridge, and you can use particles up to 300 microns. So you can use organoids in there, for example, and it stays within the consumable. So if you're using a biosafety level, let's say biosafety level 2, as long as the lab is biosafety level 2, it's not going to contaminate the outside environment. You don't need like a special air handling system like you do for a fax sorter. And you don't need any sort of markers or beads or anything. It just interrogates the cell as it is. And so how this compares to a workflow that's similar to like a fax, right? So fax sorters are notoriously difficult to use. Very often you need a specialized technician to run the fax sorter for the Institute. It takes a very long time. So it can take, 2 hours to process a sample. And there's a lot of individual steps. And every time that you touch a sample, pipetting, washing, spinning, centrifuging, resuspending, every time you manipulate it, you can introduce bias, or you can harm your cells. So a conventional fax workflow, it's about 10 handling steps, takes 2+ hours. You have to manipulate the cell, you have to tag it in some way, label them. The Levicell doesn't need any of that. So it's 3 steps. You resuspend the cells, you load it in the system, you press a button, and then 20 minutes later, you collect the cells out the other side. So this is what a typical workflow, what a typical levitation run looks like. So these are aged Jurkat cells. We use aged Jurkat cells as a model for cells that some of the population is undergoing apoptosis. So we can look at live cells and dead cells. So when we first load the cells into the system, they are in this pretty heterogeneous mix. And then as they levitate, the live cells will move to the top of the channel, and the dead and dying cells sink towards the bottom of the channel. And then we can select, before we collect, we can select where in the channel where we want to collect, which cells we're interested in. And so at the end, we have this equilibrated cell population, and the top of the channel moves to our collection channel, and the bottom of the channel moves to the waste. And then we can use the cells, the output cells, for a variety of different applications. But what do we usually get out of the system? So depending on the number of cells you put in, it doesn't matter, right? We still get high viabilities. So putting in 1 million cells to 20,000 cells to 1 million cells, we still get high viabilities. So here we have, in the black bar, the black dots are the input before Levicell, and our viabilities are 20 to 30%, and those can get enriched into the 90 percentiles. Okay, and then our yields of these cells, we get out from about 50 to close to 100% of the input that we put in. So we can routinely only lose about 30 to 10 to 30% of the cells that we put in. So depending on, and we can do this with many, many different cell types. So THP1s are a leukemic cell line, and we get very high viabilities, very high yields. PBMCs are a bit more complicated. There are multiple different cell types in a single sample, and we still get very high yields and viabilities. We can even get more complicated. So we can take a dissociated tumor sample and put it in our system, and we can get high yields and viabilities out of there. We can take, so this is a bladder tumor, then a colorectal tumor. The bladder tumor, we get much higher yield than a competitor bead sorting system. So as a proof of concept, what we did is we took Jurkat cells and that were cultured in, and we either looked at, did bulk RNA-seq of replicates without using the Levicell system, so just spinning them down, resuspending, making RNA, or we used the Levicell to enrich for live cells. And what you can see here is that when you compare the gene expression profiles of the control versus the Levicell, there's no change. Very, very little gene expression changes when we levitate. So not only are we getting the freshest, best quality samples, but we're also not changing them at all. And very often in some of these other methods, you can perturb the cells in some way that increases a stress response. And we don't see that in our system. So that was a little bit of an introduction to what the system can do. Now I'd like to talk to you a little bit about some applications. So one application that we're really excited about is using Levicell in line with single cell RNA sequencing. So one of the largest single cell companies right now is 10x. We use their system as a proof of concept to how our Levicell will perform in the 10x system. So the most important part of any single cell experiment is the sample quality. And so there are a number of different methods that people use to get the highest quality sample, whether it's gradient centrifugation or magnetic bead sorting or using a FACS sorter to get the highest quality sample that you want. All of these sample prep methods require some sort of alteration. So you need to label the cells or mark them in some way, or it damages the cells. So using high pressures or high speed centrifugation, you're putting these forces on cells on your sample, and that could perturb your gene expression profiles. And so also, as I mentioned before, the starting quality of primary samples that are coming from a hospital or coming from a freezer study, the sample quality is very, very low. And so for sequencing, you need very high viability measurements, and you're starting with very low. So other aspects of sample quality that is often overlooked is if you have clumps of cells, you tend to get multiplets in your single cell, so you're no longer looking at single cells. If your cells are damaged in some way, they can leak nucleic acids to the background. They can you can have RNA degradation. Your samples can be changing. So our system fits into the workflow to enrich for the best cells. And so when we have, I don't know how many of you are really familiar with the 10x workflow, but I'll go through it really quickly. So you start with a cell suspension, and you emulsify your cells with gel beads. And these gel beads are each barcoded that have a unique molecular identifier as well as a barcode and a poly-DT. So your 3' RNA anneals to the poly-DT from the poly-A, and then using Illumina sequencing, you can interrogate the 3' ends of the transcriptome that are then recorrelated to a single cell by use of the barcodes. So you mix the cells and the enzymes together with these barcodes, and you get single cells encapsulated in these beads, which are then cDNA is created in this emulsification, so each cell and each a transcript is correlated back to each cell. And then you sequence, and you can use various data analysis and data visualization to understand what is going on in your sample at the single cell level. But again, how do we get to a perfect cell suspension? Especially because 10x requires 90% viability for their samples. And this is actually a protocol from 10x sequencing. In their own words, they use a competitor on this in this protocol, and they're only able to obtain up to 85% viability. And so even they can't get to the percent viability that they are requiring. So what do we do? Okay, so we took early and late, both early stage bladder cancer specimen and late stage bladder cancer specimen, and we loaded the same number of cells, processed in parallel with the Levicell and just conventional processing by centrifugation. And then all work, and this was done with a collaborator called Discovery Life Sciences. They are a biobank, basically, that also does some genomics work. So if you look at our cells, so the early stage bladder cancer, looking at the Levicell, we were able to identify many more cells with many more genes per cell. So we basically we got almost 6-fold more genes per cell. So that's 6-fold the information content. And then even in the late stage sample, which actually was a higher quality sample, we still were able to improve it. We got almost more than double the number of genes per cell. We were able to identify a number of more cells. And all of this leads to better data quality. So in looking at some of these metrics, this first graph is looking at the number of reads that are in the cell. So post Levicell compared to pre-Levicell. So after Levicell, we have we have many more reads in each cell, right? So that, or a higher percentage of reads in each cell, which means that we have less background contaminating RNA in the sample. And then we also detect more genes per cell, so, or more total genes detected. So we're getting more bang for the buck with the sequencing. Okay. This is a little bit counterintuitive because in this case, we have more reads per cell. But if you think about it, we're getting more reads per cell, but we're not getting more genes per cell. So basically, a lot of these reads are throwaway trash. Whereas when we have the leve-cel, we're getting higher quality sequencing, and we're getting more cells detected. We're also getting more unique molecular identifiers. So we're getting more individual genes, more information content for each cell. So in that case, we're able to identify maybe some aspects of biology that we wouldn't be able to identify using just conventional methods. So one of the graphic outputs that we typically look at from single cell sequencing are these T-SNEs. They are projections of multi-dimensional aspects of each cell. And so it's using graph-based clustering algorithms to cluster cells as they are more like the other cells in the same cluster. And so, for example, this orange cluster here, the cells in this cluster are similar to each other. The blue cells here are similar to each other. And so when we run the leve-cel system, we get more information content compared to the conventional sample prep. So looking at the early stage, as I mentioned, we have very high UMI counts, which corresponds to very high gene content for each of these cells in this main group of clusters. So we can dig in deeper into these cells and identify maybe biomarkers of something interesting, or some aspect of this early stage colorectal cancer. Whereas the conventionally treated, there's a lot less information content here. And so our ability to analyze this data is increased by using the leve-cel, because we get more information out at the end. Looking at the late stage, we see similar differences, although the differences between the pre-leve-cel and the post-leve-cel are less vast, because the input material was in better condition for this late stage colorectal cancer. So even a good quality sample is still improved by the leve-cel. So just summarizing this part of the talk, there's the early stage sample without the leve-cel performed pretty badly. The leve-cel improved it. We got better UMI counts per cell, so more information content. We got the T-SNE plot revealed very few clusters of cells with high UMI counts in the initial sample, but the levitated sample had many clusters with high UMI content. So we're able to really identify interesting aspects of biology in those clusters. And then the later stage sample, even with the good quality as input, we were able to improve it more. So we're able to get more genes, more cells detected, and improve the data quality in both samples. Okay, so now I'd like to change gears a little bit and talk about some non-sequencing work that we do with a collaborator named Ernesto Diaz-Flores at University of California, San Francisco. He is in the Department of Pediatric Oncology. And so he's a research scientist that's very interested in leukemias and blood cancers from pediatrics. And so he undertook a study where he used CRISPR to edit the NALM16 cell line. NALM16 is an ALL, acute lymphoblastic leukemia. And so he's interested in studying oncogene addiction and mechanisms of mechanisms of drug resistance in leukemias. And so one of the genes that he studies is BCL2. BCL2 is an important gene in apoptosis. So it's a when the BCL2 is inhibited or knocked down, it pushes cells to apoptosis. So that leads to a paradox where you're interested in studying the cell in the absence of BCL2, but since the cell is addicted and requires BCL2, it's very difficult to study the cells because they die. So what he did, he used CRISPR to knock out BCL2. So he has a CRISPR construct that has the guide RNA against BCL2, and it also expresses GFP. And so he takes the NALM16 cells and he transfects them with the BCL2 construct, and then he FACS sorts and obtains all the GFP positive cells. So if they're GFP positive, that means they're expressing the CRISPR knockout. And then what he does is he cultures them. And so if he cultures them directly, the dead and dying cells that that have BCL2 knocked out, they basically poison the culture and kill the cells that are surviving because of cell-cell signaling. So the cells would routinely die if you take these cells and just culture them directly. So what he did was he took the GFP positive cells and then put them through the levicell. And so he was able to purify live cells that were BCL2 knocked out, and then he can study the mechanism of cell survival in the absence of BCL2 in a cell line that's BCL2 dependent. So what he first did was he just took his NALM16 cells and he put them on the levicell before changing them at all, before editing them with CRISPR, to get the most viable NOM16 cells possible. So his culture of NOM16 cells were at 55% viability. He then put them on the Levicell, and he was able to get cells that were 85% viable, 84% viable. And then the bottom channel, the waste channel, he's only wasting 22% viable cells. So most of the dead cells are going to the bottom channel. Most of the live cells are going to the top channel. And then he uses those top channel cells. Oh, 1 second. So this just shows like the difference in the starting viability. So his input, the starting viability, very low viability, a lot of cells that are apoptotic, a lot of cells that are necrotic. And then his final viability, looking at the top cells, he has very highly viable cells, okay? And then a lot less of them are apoptotic or necrotic, whereas the waste cells, most of them are necrotic or apoptotic, very few that are highly viable. Okay, so he took these cells and he used CRISPR, and as I mentioned before, he was interested in BCL2, but he did some control experiments as well. So he also knocked down CDK9, he knocked down MCL1, and knocking down CDK9 or MCL1 didn't kill the cells, right? So he still had 90% viability from CDK9. He had high viability from knocking down MCL1, but when he knocked down BCL2, he only had 8% viability. So what he did was he took the output from this gate, from the FACS sorter, and put it into BCL2, into the Levicell. And so he was able to take this dead population, right? This population, and he was able to enrich for viable cells. And so before Levicell, he only had a maybe 50% that fell into this gate, which maybe only a very small population of that is actually viable. And then taking that viable portion out of the Levicell, he was able to enrich for over 75% viable. And you're doing a Western blot on these cell populations, he was able to see that BCL2 was in fact knocked down. The GFP control is present. So these cells are now able to be used in downstream functional assays for understanding BCL2's role better in the cell line, and then ultimately being able to understand the mechanisms of drug resistance and BCL2 dependence in leukemias. One very, very interesting aspect that we noticed is that his live population actually, once he knocked down the BCL2, his live population formed these two different populations. And so Ernesto's in the process of understanding this more, separating these cells further and functionally characterizing them. So this is all work in progress. He's also very interested in making patient-derived xenograft models and spheroids. So he uses the Levicell to enrich for very high viability spheroids, or taking a pheresis from a patient and enriching for very, very high viability. And then he can put million cells into each naked mouse, SCID mouse, so he can develop a patient-derived xenograft and then study what's going on with these patients and what cells are expanding and are protumorigenic in the mouse. So in conclusion, the Levicell system is able to obtain high yields and viabilities without altering the sample. It simplifies and streamlines 10x workflows. We're able to enhance CRISPR screens and knockout experiments, allowing a user to enrich for cell populations that would otherwise be lost or not able to be studied. And we can also enhance patient-derived xenograft workflows and development. So with that, I'd like to thank the team that worked on this, especially Ernesto at UCSF. And I'd be happy to take questions. If you're interested in learning more about the system, please visit the website, levitasbio.com. You can send us an email. And we'll be happy to chat with you more about anything. So thank you very much. Yes.
Thanks a lot for this very, very clear presentation. And I think this is the system that you have just presented is, I think it's way better in number of applications, even comparing to the FACS, which is I'd like to learn more about it. But now I'd like to ask Dr. Ahmed Abdel Mawdoud, the program director, the program coordinator, the biotechnology program coordinator here at the Basic and Applied Sciences. You have a question? Go ahead, Dr. Ahmed.
Okay, well, it's very, very interesting. And it's interesting that you are using a microfluidic system. We just started this research, I mean, 2 years ago. Okay, now for the separation, the cells, the dead from living cells, is this including inertia? I mean, could you elaborate as well on the way how would you separate this one, or this is proprietary to the company?
So I'm not a physicist, but I can give you a little bit more detail. So the cells are resuspended in a buffer containing an agent that's similar to the agent that's used in MRI. And so this is a paramagnetic substance, and it is excluded from live cells, but dead cells take it up. And so if you imagine a beach ball, if you have the ball in water, the ball is impenetrant to the water, and so it floats. But if you poke holes in the ball, which basically is what's happening when your cells are dying, they're making holes in the membrane. And so the water can seep into the ball and it weighs it down, and it makes it less buoyant. And so the highly buoyant live cells are resuspended in this system, in this media, that is then allows the cells to experience only the buoyant force to separate it. So it basically, it's almost similar to like it negates the forces of gravity almost, right? So the cells are buoyant. The cells that take up the media, the levitation agent, become weighed down, and they sink in the channel.
How width is your channel? The channel
How
for
large?
the
How large is it? It's
yes, the width and the depth.
Yeah, I believe it's 2 millimeter square. So it's not quite microfluidics.
So this is a continuous system, yeah?
Yes.
Okay, thank you. Thank you very much.
Yes.
I just have a question regarding this. So now I do have question from Dr. Ahmed Abdelmenem, the Dean of the school. Is not here.
Ready?
Okay, so let's get back to my question. So now I can understand based on what you are explained right now, the method of the separation of the cell. But when you're talking about the CRISPR, you isolated two different cell populations.
That last image where you
Yes.
had two in the live.
Yes.
So
is
how
it-
do
is
you-
it just- how the technique would be able to separate two different kinds of a living cell with different, because this is very clever, by the way, and
Yeah.
it's very important to separate two separate populations, and the separation pretty much is very clear.
Yeah. So this is a great question. So there are sort of two ways you can think about it, of how to do it. One way would be to deplete one of those, with some sort of depletion agent. Another way, but if you're interested in actually collecting them separately, you can take the first pass through the Levy-cell system, and then you collect that output, and then you put it in the Levy-cell system again, and then you collect the two, you take the split in between those two populations, and then you can select one in the top channel and one in the bottom channel.
Okay, got that.
So although we call the bottom channel the waste channel, because of all in most applications it is a waste channel.
But you can collect
for that application, you could force one of the populations to the waste channel and still collect them.
Okay, got that. Yeah. So I think my next question would be for Kiara about the expenses of the system.
Yes.
And you have,
can you hear me?
Yeah, I do hear you. How you doing, Kiara?
All right. So what we currently sell is the Levy-cell platform. So this is the capital equipment. And then we have the cartridges and the levitation agent as consumables. International list price for the Levy-cell is $100,000. So this is the price of the capital equipment. The price per run, so each cartridge and the related levitation agent that you need to use per each run is around $100. So that's the consumable price. So we sell like 10 cartridges, like a package of 10 cartridges together for around $1,000. So yeah, and the instrument comes with standard warranty. It's a plug and play instrument, so it doesn't need installation. It doesn't need a service engineer. It doesn't need maintenance. It's a benchtop instrument. So yeah, this is what it includes. Also, it comes with a computer next to it that you use to operate the system with.
Perfect. Do you have a representative in the Middle East or?
Not yet. So we started to build our commercial organization this year. So I've been appointing distributors all throughout Europe. We have a distributor in Qatar, but we don't have yet a distributor that covers Egypt or
so you have one in
other.
Qatar?
Yes, we do.
Okay.
I don't know if that would be helpful for you, but we can, we can continue the conversation if you want to, in terms of how to find a local partner that can support you. Maybe you can also give me some suggestions on who you normally work with.
Sure. So thanks, Kiara. And
you're very welcome.
Thanks, Taz, for this clear, informative presentation. The technology was I really like it, and I'm sure that it's gonna expand in the near future, comparing to the facts with all of his long procedure. And again, comparing to the facts also from the graphs that you presented, what I really liked and I got impressed, the number of cells comparing to the number of genes also, which was very very impressive for me. I'd like to thank you, Seth, again for giving us this presentation. And also, I'd like to thank you also for me seeing you after a long time. And hopefully we'll be able to meet again once in the near future.
Yes, absolutely.
Thank you, Seth.
So good to see you again. Thank you, everybody, for your attention. And contact us if you have any further questions.
Sure.
Great.
Thanks,
Thank you very much.
Thank you. Goodbye. Thanks,
bye now.
Seth.
Enjoy the
Bye.
rest of your day. Bye-bye.
You too. Bye-bye.